Hi everyone, welcome to my channel Pure Biology, and today I come with another video, that is the Lytic cycle. Okay, so before going to this topic, I have to say sorry because I didn't give any video for 1 month, last 1 month I think so. So that's why I say sorry to you, to my viewers, and today I come with this video. This is related to genetics, obviously. The not genetics, you can say that obviously the bacterial genetics. Okay, so generally the bacteria, they can reproduce in the two way. Bacteria means the phage virus, not bacteria, you can say the phage virus. So here in this series I will try to explain about the bacteria and the virus related topics. Okay, so this is the Lytic cycle, and you can say this cycle is the reproductive cycle of the phage virus. Okay, so generally two kinds of life cycle or the reproductive life cycle, these are related to the phage virus. One is the Lytic cycle and another is the lysogenic cycle. So this Lytic cycle, generally it is seen in the virulent phage. Okay, virulent, one thing, this is seen generally virulent bacteriophages. Okay. So what is the term is the virulent? That is the virulent means the bacteriophage. These are the harmful. Okay, or phage virus. These are the harmful. Harmful means when it attack any bacteria, they try to destroy or damage this bacterial cell. Okay, and another is the antivirulent. Here the phage virus is not harmful. So okay, so in the next series I will try to about this topics, that bacterial cell and the viral cell, okay, or phage virus. Now this is the topic, this is the Lytic cycle. So Lytic cycle is what? Come to the point. This is the Lytic cycle, which is called the Lytic cycle. This Lytic cycle is the lysis of the host cell. Okay, this is the lysis of the host cell and the release of numerous viral progeny. Clear? So that means lysis, mean from this word lysis, this Lytic comes here. Clear? So here the first bacterial cell is attacked by any phage virus, and after that, after completion of the life cycle within the bacterial cells, the damage of the bacterial cell is seen here. That means the lysis is seen here. Lysis of which one? This is the host cell. The lysis of host cell that release the numerous viral progeny. So here I just told about you the virulent bacteriophages. The bacterial viruses exhibiting Lytic cycle only are also known as the virulent bacteriophages. And these because why these are called the virulent? They generally cause the death and the destruction of the host bacteria. That's why these are called the virulent bacterial cell. And here this Lytic cycle, this consists of the 5 stages. One is the attachment, second is the penetration, third one is the synthesis, fourth one is the assembly, and fifth one is the release. Okay. So what happened in the first case, that is the attachment. Here first the bacteriophage, these are attached with the phage virus. Okay, that means the phage virus first attached to the specific host cell. These are known as the receptors, or you can say that phage virus, these are attached to the bacterial cell wall. This is the first point. Second one is that what? This is called the penetration. That means entrance of the phage virus within the host cell. Okay, just after attachment, they try to enter their genetic material or the DNA within the host cell. This is called the penetration or entrance of the DNA. Third one is the synthesis. What is this? When the bacteriophage genome that enters within the cytoplasm that try to bind with the host nucleic acid, okay, and with the protein synthesis apparatus, they initiate the synthesis of the viral protein and DNA. Again I repeat, what is happening here, the phage viral DNA when enters within the host cell, they directly attach with the host genome, and with the protein synthesis of the host genome, viral proteins and DNA also be synthesized. Clear? And as next point, that is the assembly. What is this? As the viral proteins are synthesized, they self-assemble into viral components such as head that containing DNA, tail and the tail fibers. So what is happening here? In the first, when the synthesis process is occurring, in that situation viral proteins and DNA form. And these viral proteins DNA, once they are formed, then the another part of the phage virus, that is the head, tail and the tail fibers, these are already formed within the bacterial cell. And here the DNA that present within the head of the phage virus. Okay. And just after completion of the synthesis or assembly process, after just completion of the assembly process, the formation of the numerous intact phage particles within the cell. That means after just completion of the synthesis and the assembly process, number of phage viruses, that means the large number of or numerous phage virus, these are formed within the cell. Okay. And after assembly step is completed, okay, this point is also included within the assembly step. Okay, just after synthesis, the formation of the numerous number of the phage virus, these process also included within the assembly process. And just when the assembly process is completed, that viral proteins cause the lysis of the host cell. This is the process where the release of the phage virus is seen here. Okay, just after completion of the assembly process, that means the numerous number of phage virus when totally formed within the bacterial cell, then they try to damage or destruct the bacterial cell to release themselves. Okay. Generally, which is known as the burst size, the average number of the phage particles that produced by each infected cell, this is called the burst size. That means within one cell, average number of phage particles are produced. How much number of phage particles are produced? This is called the burst size, and this number that ranges from 50 to several hundred. Clear? So this is the process of the Lytic cycle. This is very easiest process of the life cycle process of the phage virus. And after extension of this, I will try to explain the process by a rough drawing and rough diagram, obviously. And obviously I will try to explain this with a graph. Okay, so continue with this. Okay, here this is the rough diagram. First one is the attachment, second one is the penetration of the viral DNA, third one is the synthesis of the virus nucleic acid and the protein, and fourth one is the assembly and the packaging of the phage virus, and the last one is the release or the lysis. And I said one wrong words, that is the viral DNA attached with the DNA of the host cell. No, this is not possible here. The viral DNA themselves produce their own DNA by the process of replication, not attached with the host DNA. Okay, so I have a mistake. Okay, so this is the process So in this process, here the host cell, they actually by the process of replication, they produce their own DNA, and here after entering within the host cell, the viral DNA, by the process of replication, they produce their own DNA individually. Okay, here no attachment with the viral DNA and the host DNA is not seen, okay, or is not occurred. And this is the graph where these phases are. First one is the active period, second one is the latent period, and the third one is the intracellular viruses, where the most intracellular viruses are produced. Okay, and the first phase, this phase is called the viral absorption and entry. Second, this eclipse period, here the viral replication is possible, viral DNA replication is occurring, and the third phase, here the viral maturation is seen, and the fourth phase, here the viral release is seen. Okay, or release of numerous viruses that are seen here. Okay, so here this is called the one-step growth curve, and this is the representation of the overall change with time in the amount of infectious virus in a single cell that has been infected by the single viral particle. Okay, that means here one cell is infected by one phage virus, and from this one phage virus, numerous phage viruses that are formed within one cell, and the release of this numerous phage viruses are seen. Okay, and this procedure is actually this is represented by this curve, or you can say this growth curve, and this is called the one-step growth curve. Clear? So here are the periods. One is the eclipse period, second is the latent period, third one is the mature phase. Okay, latent period, that remains within this and this. So what happened in the eclipse period? Here no variants can be detected within the cell. You cannot detect any cell, any phage virus within the cell. This is the first point, or this is the eclipse period. Second is called the latent period. Here the mature phage that form. So here no extracellular variants can be detected. So here that was no variants can be detected. Here no extracellular variants can be detected. And the last phase, this is called the mature phase. Here the that accumulate intracellularly until these are released. So this step or this position, this is called the actually this is included within the mature phase or mature period. So what is happening here? Maximum number of phage viruses that are accumulated in this period, okay, or in this situation. Clear? This is included within the maturation phase. So this is the actual growth curve of the virions or the phage virus, which explain that when the viral absorption and entry is occurring, or then replication is occurring, or the maturation is possible and the release is possible. That means you have to understand that before releasing the phage viruses, the intracellular viruses are all accumulated. Okay, and you can detect in that moment the viruses present within the bacterial cell. So the Lytic cycle is completed here, and I will try to explain the lysogenic cycle in my next video. Okay, so please you continue with my channel. Thank you.